Hello, everyone. My name is Erin Newburn, and I'm the director of the Field Application Scientist team at Personalis. We are very thrilled to be a part of this year's SITC meeting. I'll be presenting a talk entitled Leveraging Comprehensive Genomic Data for Diagnostic Capabilities and Composite Biomarker Discovery in Immunotherapy. Back in 2013, Chen and Melman described the cancer immunotherapy cycle. And the cancer immunotherapy cycle involves the coordination of numerous factors, both inhibitory and stimulatory, and there are a variety of potential therapeutic targets currently under clinical evaluation that are targeting these various steps in the process. And cancer immunotherapy continues to make great strides, and the field has been revitalized in recent years, but unfortunately still a very small percentage of patients have a durable clinical response to immunotherapy treatment. Checkpoint inhibitors such as anti-PD-1 and anti-PD-L1 are effective in just 10 to 30% of the patient population. So there are still a variety of clinical trials that are working with a combination therapy strategy. And so as the search for more accurate biomarkers to understand immunotherapy patient response continues, we see multiple groups and multiple investigators that are expanding beyond the metrics currently in clinical practice to other key parameters, such as, for example, developing expression signatures from the tumor material, taking a finer look at the patient's HLA-related genetic variation, understanding the antigen presentation machinery, or looking at the TCR dynamics that are coming from the tumor material. Again, all with this ultimate goal of identifying biomarkers that have stronger predictive value. And obtaining patient samples is critical. These are very precious patient samples, and it's often difficult to obtain material for patient response prediction. Traditional processes have often required multiple samples to perform multiple assays, multiple technologies for biomarker evaluation. So for example, perhaps having to send a patient's blood sample out to understand their HLA type, maybe sending then a tissue section to look at the mutational landscape, perhaps another tissue section to develop expression signatures, and maybe another tissue section to understand the MSI status. And the result is that investigators may have difficult decisions. Perhaps there's a trade-off about which particular assay they can run when sample is limited, but also working with complex data integration when you're working with various assays, various technologies. At Personalis, we have put forth a much more efficient process. We perform a dual extraction of both DNA and RNA from the tumor material. We then perform a very unique next-generation sequencing library preparation, and then process the data using our ImmunoID Next platform to put forth a very data-rich set of bioinformatic output. ImmunoID Next, the product vision was to really use this as a singular solution, to use a single sample to consolidate multiple biomarker assays into one. And our product was developed to enable a comprehensive characterization of not just the tumor genomics, but also the immune repertoire and the tumor microenvironment. And we've maintained a sample sparing approach. We can work with just as few as 5 FFPE slides in our process, which we then interrogate both the DNA and the RNA features for biomarker evaluation. This is a scalable approach. It enables the identification of today's biomarkers that are in clinical practice, such as MSI status or TMB, but it also enables a path for more novel immuno-oncology biomarker identification as well. And the platform itself is powered by a very innovative assay design. First, by the depth of sequencing coverage that we provide, but also our proprietary augmentation and enhancement. So we perform a 300x mean whole exome sequencing coverage of the tumor. This is also paired with 150x coverage of the patient's normal germline sample. But our platform utilizes our proprietary ACE, or accuracy content enhanced technology, to augment those difficult to cover genomic regions, such as GC-rich regions that are often missed by a typical off-the-shelf kit. And I'll speak a bit more to this on the next slide. This is paired with our enhanced transcriptome sequencing of the tumor, We deliver 200 million total reads, and we use a capture approach in our enrichment method. We utilize those same ACE probes that we've used for our whole exome approach to tile across and enrich for the messenger RNA. Secondly, what we've also done in our assay design is we have provide boosts of ultra deep sequencing coverage in critical areas important for oncology and precision oncology. The first being a set of 247 cancer-related genes associated with solid tumors, as well as hematological malignancies that we boost to greater than 1000x coverage. This enables diagnostic capabilities of this very critical clinical content. And we can also provide a clinical report to an ordering physician on those 247 genes. Other areas that we've enhanced include the TCR and BCR gene regions. That allows a look at the TCR dynamics, another very emerging immuno-oncology biomarker. We've also enhanced the HLA loci, and our platform can deliver a variety of HLA-related analytics, including HLA typing, somatic variant calling for the HLA genes, and also HLA loss of heterozygosity reporting. We do also report on the MSI status of the patient's tumor and can also detect 7 of the most common oncoviruses with our approach. So as you can see, this really is a first of its kind approach. It's been very carefully designed with enhanced sensitivity and specificity, but also to enable investigators to look at not just the tumor, but also the immune-related components of the tumor microenvironment in a single assay. And as I mentioned on the previous slide, our platform is powered by our ACE, accuracy content enhanced technology. And the issues with the typical kind of some of the standard off-the-shelf sequencing methods is that they often have gaps and inconsistencies in sequencing coverage. And that's demonstrated here on this plot. So what I've done here is just taken a typical gene. This is actually STK11. And what we're showing here is if you're running a standard whole exome sequencing approach, so this is sequencing, depth of coverage on the y-axis, and we've just concatenated all the exons so that you can look at gene-wide coverage across this gene. And what results is what I refer to often as peaks and valleys, perhaps areas very sufficiently covered by the technology, but you can have gaps and often completely areas that are devoid of sequencing coverage that might be missed. And this is what we've recognized and addressed with our proprietary ACE chemistry. So this is now a nice visual of the quality of the data that results from our enhanced whole exome sequencing approach, the blue plus the green, where we have strived here to give you much more uniform coverage across the entire genomic footprint so that you can have an accurate assessment of both neoantigens, mutational landscapes, as well as other biomarkers. And so the landscape of predictive biomarkers in immuno-oncology continues to evolve and develop. And there still is an existing need for a more robust patient stratification of response. And establishing accurate biomarkers for immunotherapy have been elusive due to a variety of complex mechanisms. Some particular considerations are listed here on the slide. For example, the need for more cancer indication or subtype-specific thresholds for these biomarkers, having a better understanding of the patient's prior treatment history and the impact of that on the biomarker, and also understanding the importance of a patient's HLA genetic variation. So there's really growing evidence that likely a single biomarker will not be sufficient in identifying patient response in immunotherapy. In the past, with targeted therapies, the approach has been a single gene, a single analyte, but a more integrated multi-dimensional approach is needed to stratify immunotherapy patient response. And so in our publication back in August in the Journal of Clinical Cancer Research, this publication illustrates how a high content, very information-rich approach that we have created with ImmunoID Next, both the assay and the informatics, can be utilized to better understand patient response to immunotherapy, but also to further evaluate immune-related resistance mechanisms. And so in this publication, we utilized a cohort of 51 metastatic melanoma patients that had been treated with anti-PD-1 therapy at the Inova Shar Cancer Institute in Virginia. We received those pre-treatment FFPE tumor samples along with the patient's blood sample, and we ran our broad immunogenomic profiling assay of ImmunoID Next. And then we're able to take the clinical response information in conjunction with the molecular data to further evaluate biomarkers of therapeutic response. And so we first looked at the mutational landscape of the cohort as tumor mutational burden is a key biomarker in melanoma. In our platform, TMB is optimally calculated by using the whole exome footprint. This is as opposed to, as many of the smaller gene panels will utilize several hundred genes to bioinformatically extrapolate the TMB. We utilize our whole exome footprint, so looking across the exome to report out on non-synonymous somatic mutations, both SNVs and indels per that genomic footprint. So what we found in our cohort is we observed a slightly higher tumor mutational burden in responders and compared to non-responders. However, there was overlap between the groups, suggesting in our cohort, this did not seem to be a strong predictor of response. And this may be in part to the level of TMB variation that we saw in different melanoma subtypes, as well as tumor site locations. And that's what we see here on the right panel. We also, as a next step, computed the neoantigen burden from the tumor materials. So looking at the potentially more immunogenic variants. And this was done using our internally developed neoantigen ID pipeline. With our neoantigen burden assessment, our pipeline predicts neoepitopes using Sherpa. This is our proprietary machine learning tool for MHC class 1 presentation. It was trained using very clean immunopeptidomics data sets at Personalis. So our pipeline predicts both class 1 as class 2 neoepitopes and provides a neoantigen load, neoantigen burden assessment. And when we calculated our neoantigen burden in the cohort, we found that it was more strongly associated with response, as well as progression-free survival with more significant p-values than TMB. We did observe some unique profiles in our cohort that we wanted to characterize further, and I'll highlight a few of those cases next. So this first example was a partial responder, only partially responded to anti-PD-1 therapy, but had an elevated neoantigen burden. And we looked at multiple parameters within our output to try to understand this patient's reduced therapeutic response. And we found that alterations at the HLA level may play a role. We did find from the HLA typing results that this individual was heterozygous for all 3 of the class 1 genes. However, when we dug into the tumor analysis, we found a chromosomal deletion that resulted in the removal in 1 allele in all 3 of the class 1 genes. So this HLA loss of heterozygosity event is potentially detrimental to the number of neoantigens that could be presented to the immune system during immune surveillance and may explain their partial response to treatment. We also were curious about another patient. In this patient, again, a relatively higher neoantigen burden, but did not respond to anti-PD-1 therapy. And so we found in this patient 2 very damaging somatic HLA class 1 mutations. The first one, HLA-A0201, was a stop gain mutation. The second mutation, HLA-B1501, was a splice region variant. They were both found at a high allelic fraction and had a high variant effect impact. And then when we looked at the full list of predicted neoantigens coming from this patient, we actually found that 40% of those predicted neoantigens were predicted to bind to these mutated alleles. Again, this could be a potential mechanism of tumor evasion and may explain the lack of response in this case. In another patient, a non-responder, we observed a somatic frameshift variant in beta-2 microglobulin. B2M is a very integral component of our antigen presentation machinery, and this frameshift variant perhaps limits correct folding of the HLA and ultimately antigen presentation of the peptide MHC complex. And this is very consistent with other studies in the literature showing a similar mechanism of resistance. So as you can see from our approach, we can report on tumor mutational burden. We can also report on neoantigen burden, But what we can also do is integrate key molecular features to create a more composite score, composite biomarker. And when you're counting non-synonymous somatic mutations up with tumor mutational burden, that's a first step. And then as a next step, looking at the more immunogenic variants with neoantigen load, basically counting up neoepitopes. The question then might be, how relevant are these neoepitopes when you're assessing tumor foreignness if they're not able to actually be presented to our immune system for surveillance? So we wondered that if we took into account the patient's HLA status, their impaired neoantigen presentation in conjunction with the neoantigen load assessment, would this be able to better parse out responders and non-responders? And this is what we did with our creation of NEOPS, our composite neoantigen presentation score, where we utilized our neoantigen load assessment as the foundation, but then took into account impaired neoantigen presentation. And what we found is that when we utilized our composite biomarker of NEOPS, this biomarker was more strongly associated with overall response as well as progression-free survival. So by integrating antigen presentation features into this composite biomarker, it was able to strengthen the association when we looked at anti-PD-1 response. We also wanted to evaluate the transcriptomic profiles of the patients to determine if transcriptomic features were also associated with immunotherapy response. So from our RNA data, we profiled and analyzed the TCR beta repertoire diversity using our platform, and we found that TCR beta clonality was significantly associated with overall response as well as progression-free survival. Also from the RNA data, we did characterize the immune cell populations within the tumor microenvironment. And this data showing here was done using publicly available gene sets to produce this semi-quantitative immune infiltration score. And we found using this approach that both the responders and non-responder patients shared similar immunocellular distributions. And that's what you see here, the responders here shown in green, non-responders shown in gray. So in summary, from our recent publication, we found our composite biomarker NEOPS achieved greater separation when we looked at responders and non-responders compared to single analyte, single genes, or tumor mutational burden signatures. And when we accounted for tumor escape mechanisms and integrated them into this composite score, it gave a deeper representation of tumor antigen presentation. In addition, NEOPS can also be clinically feasible. We were able to perform this broad, comprehensive profiling using a very limited amount of tumor material. So there are many studies out there that are also leveraging the power of RNA-seq data to try to understand if this can be a powerful tool to better understand immunotherapy response. And as another example of this, this article was published in Nature Medicine from the Javelin Renal 101 trial. This is one of the largest renal cell carcinoma cohorts of its kind. And in the study, the investigators found significantly prolonged progression-free survival in first-line avelumab plus axitinib versus a multi-targeted tyrosine kinase inhibitor. In this cohort, when they assessed tumor mutational burden or PD-L1 expression status, it was not associated with progression-free survival. However, they were able to utilize our Personalis transcriptomic data to develop a expression signature. This was a 26 genes that they utilized. These genes had immune-related functionality, and by developing this signature, it was associated with progression-free survival. Again, highlighting here the power of transcriptomic data, how it may be able to be used to better evaluate patient response. And historically, FFPE materials have been quite challenging for RNA-based next-generation sequencing. Due to the chemical modification process with FFPE, this often results in degraded more fragmented materials. Methods such as capture enrichment protocols can allow you to overcome some of these challenges with FFPE, to work with a more degraded or a more limited starting sample material. In our process, we utilize our optimized ACE probes that target the exonic regions to enrich for the messenger RNA. And what we achieve here, again, another blue-green plot here I show on the bottom right, is basically blue plus green. We really work, as we do with our exome, with our transcriptome data, to result in more uniform coverage across the genomic footprint for more increased sensitivity. And this exome capture enrichment protocol was developed to alleviate some of the challenges with working with FFPE. However, it's applicable for various sample types. And I should mention, in our lab, we receive a variety of different sample starting materials: FFPE, fresh frozen, whole blood, PBMCs. On the left here are just a few snapshots of some FFPE materials that the lab has received. And as you might know, these come in a variety of quantities and qualities, and this is frankly the reality of working with clinical FFPE materials. However, in our lab, we have really optimized to work with more difficult, more challenging starting materials. And what you see here on the right, a quick graph to show, is that regardless of the sample starting material, if it's a fresh frozen material, if it's archival FFPE or more freshly prepared FFPE, we have a very high percentage of success rate of being able to extract both DNA and RNA from those materials. So in short, our operation team is highly skilled in working with a variety of sample starting materials, especially more challenging materials that have been traditionally more difficult for next generation sequencing approaches. And so in the slides I've presented, hopefully I've demonstrated how an information-rich platform may be able to give accurate and comprehensive biomarker discovery. I've shown just a few of the analyses that are capable by our ImmunoID NeXT platform. I've introduced NEOPS, our composite biomarker that takes into account antigen presentation impairment along with neoantigen burden. And this is an excellent example of a more multidimensional approach for biomarker discovery. The NeXT platform is unique in that it provides our customers with the ability to simultaneously, comprehensively characterize both the tumor and the immune-related components of the tumor microenvironment, and to do so in a very sample-sparing way. As I mentioned before, we work with just as few as 5 FFPE slides in our process. Our goal when we obtain that tumor material is to try to get to a 1 millimeter cubed volume of a tissue. We then extract both DNA and RNA, and we look at not just the quantity of materials in that assessment, but also the quality. So we look at the size fragment distribution along with the nanograms of material that we've extracted. And again, we've really worked to optimize to be able to provide a very comprehensive amount of data from very limited tumor material. So in my concluding slide, with the platform we have established, we really are a partner throughout the drug development process, throughout drug discovery. Currently, we are supporting many biopharma customers in their clinical development and translational research of their phase 1 through phase 3 trials. This all started with our ImmunoID NeXT offering, but we've also now enhanced this with our NeXT liquid biopsy offering. We provide an exome scale assessment of the patient's plasma samples. In addition to that, I briefly mentioned that we do provide clinical testing support with our NeXT DX LDT. So this is based on the tumor's molecular profile. We can deliver a clinical report to an ordering physician on the relevant therapy recommendations as well as clinical trial matches. And this is another very exciting element for us at Personalis, as it represents our first step towards a companion diagnostic. And this is precisely what we are doing at Personalis with the development of our NeXT CDX. I also wanted to quickly mention that we do offer also various research solutions at Personalis, such as various flavors, various configurations of RNA sequencing, microRNA sequencing, as well as whole genome sequencing to support the early discovery and preclinical work of our pharma partners. And so thank you very much for your attention today. Hopefully this presentation gave you some insight of how at Personalis we are really working to take on the challenges in biomarker discovery with our ImmunoID Next solutions. Enjoy the meeting.